All right. Let's see if I can get this to work. All right, you can see my screen.
Yes.
And okay. So yeah, I'm going to talk about these pulse oximeter phantom approaches and just show a little bit of some of the work that we've been doing. We've teamed up with Florida International University, Professor Ramela Roman, her group over the past 6 months to work on this initial phantom. I'm going to start out with a little more kind of background on this topic. Over maybe the past 10 years, we've done a fair amount of work at FDA on developing optical phantoms for a variety of diagnostic imaging and sensing technologies, including most recently cerebral oximetry. And we applied our cerebral oximeter phantom to look at the issue of racial bias and presented our first work in 2019. And publication was delayed a bit from COVID and some other things going on at FDA. Finally published it this last year. I'll talk a little bit more detail about it, but it's really this work that kind of also got us interested. This work was directly relevant to other work going on in the standards committee on cerebral oximetry. And we think this has relevance to pulse oximetry as well. So I think it's worthwhile talking a little bit about tissue optics and the impact of skin pigmentation on optical devices. It's well known that the absorption coefficient of melanin decays strongly with wavelength across the visible and near infrared. This is one of my favorite graphs to show about this because I think it really clearly illustrates the direct relationship between the absorption spectrum of melanin and the resulting impact of skin pigmentation on optical measurements. You get stronger decrease in detected signal at shorter wavelengths, less impact at longer wavelengths. So here we have this large impact that Mike Bernstein was talking about. Here it's showing about 80%, somewhere around there. At 940, much less. So it's a big impact, but there's a differential between the two wavelengths. We believe that had something to do with these results in cerebral oximetry that were found by UCSF about 10 years ago. And this also kind of guided some of our cerebral oximetry phantom work. So, but the impact of melanin on optical devices goes well beyond oximetry devices, bilirubinometers, photoacoustic imaging, I'll talk about a little bit later. There's a very interesting paper about bilirubinometer design and the algorithms used and how this is actually not a very unusual approach, this kind of spectral decomposition where if you have multiple absorbers you know a priori what their distribution looks like, you can extract each one of them or identify the relative magnitude of the contribution from each. And so this can be done for melanin, and you can essentially remove this slope effect and get at the hemoglobin and bilirubin content more directly. So it's, this impact of melanin is really well known, and devices have their algorithms have essentially been designed around this and to correct for it. Similarly, in hyperspectral oximetry, melanin is quantified and then essentially extracted from the optical signal to get at the underlying oxygenation levels. So there are a variety of roles for pulse oximetry phantoms that we see. At FDA, we're particularly interested in what we call regulatory science tools. So we like to see the development of standardized tools for device development and regulatory evaluation. Hopefully they can help predict trends in performance, and ideally in the long term, quantitative clinical accuracy would be a goal, but that's very difficult to achieve. We want to be able to use phantoms to study in isolation different biological factors, whether it be finger size, hematocrit, perfusion, and of course, epidermal melanin content, and even combinations of these factors like melanin content, in addition to signal strength, as to kind of replicate the work done by UCSF lately, showing that this is a particular case where there can be significant problems in high pigmentation and lower signal strength. Phantoms can be used to study the effect of environmental factors, device design, and the changes in device design and the algorithms, including algorithms that might be developed to mitigate the impact of melanin. I think Sandy mentioned earlier the idea of using phantoms as a way to test in regions that are unsafe. So in regimes like pediatric devices where the healthy human subject studies are not performed. This might be an area where we can contribute to evaluating devices, and also just generally having a better understanding of light tissue interactions that underlie any sort of racial disparities in pulse oximeters and other optical devices. Some of the other benefits of phantom-based testing include the ability to test under well-controlled conditions with known material properties. We can cover a wider range of physiological conditions, as was alluded to before. We think that phantoms can provide important information that elucidates clinical results that are not really well understood, and may eventually reduce the need for expensive in vivo studies. So it's interesting to note that some of the first pulse oximetry phantoms that we see in the literature actually were published 30 years ago. They basically have some of the same features we see in the more recent approaches, where there's a flow loop that has components to oxygenate the blood and generate pulsation that's biomimetic. There are phantoms that have turbidity and blood flow, and that these can be used to essentially mimic a clinical study in providing the measured oxygenation as a function of a gold standard like co-oximetry. But it's also interesting to note that there's kind of a gap of about 20 years where there was relatively little work done in this area, apparently, and we see more recently a number of different phantoms have kind of been developed for pulse oximetry and PPG as well. And of course, I'm glad Mike Bernstein covered pulse oximeter testers, not an area I know too much about, but certainly they're very popular for certain roles in evaluating pulse oximeter performance. So some of the shortcomings that we see of these prior phantom-based test methods, one is a lack of consideration for the true optical properties of tissue, most importantly, the pigmented epidermis optical properties, and then other tissue regions. There was one phantom that actually used a sheet of paper to simulate the turbidity of tissue, which is really not a great way to simulate turbidity. There's in general a lack of mechanical property characterization that's provided in prior papers. The geometries that have been developed typically are highly idealized, and some of them can't even accommodate clinical devices. There's typically minimal verification data provided in terms of the compliance or ability of channels to expand and the PPG waveforms that are produced. And it's almost no validation at all against clinical devices in comparison to different clinical results. And of course, we've heard about some of the limitations of electronic testers. So some of the requirements for the phantoms that we want to develop, we want realistic optical and mechanical properties, and as well as morphology, although it's certainly a question, how do we balance the desire to make a phantom biomimetic versus make it simple and or realistic to construct in a uniform, reliable, repeatable way. We want the ability certainly to adjust saturation, the percent modulation, epidermal optical properties, and a variety of other parameters. We want to be able to accommodate a wide range of clinical pulse oximeters, and generate clinically realistic PPG waveforms and saturation results. So in trying to generate a phantom that represents the human epidermal pigmentation correctly, we turn to the literature, and Steve Jacques is one of the people who's done a lot of work in this area. He's published at least 4 papers and has come up with at least 4 different curves to describe the absorption coefficient of melanin. So we've chosen kind of a average value, and we can use that to estimate essentially what the epidermal absorption coefficient would be for a highly pigmented skin. And then we use this distribution to identify tissue mimicking materials that best recreate this spectral distribution of absorption. And I should note that the melanosome volume fraction that we see mentioned by Jacques goes from 1 to 43%, but certainly for fingertip, it's going to be significantly less than that. Okay, that didn't come out right. All right, so here are a couple of the studies that we've done with pigmented epidermis phantoms. So the cerebral oximetry study that I mentioned before is kind of illustrated here. This is our phantom with a array of channels in a turbid matrix that was 3D printed, and we have these superficial layers made of PDMS with nigrosin embedded in them, and we can generate these spectral distributions that represent the absorption coefficient of the epidermis for different skin tones. And then by measuring the saturation of blood injected into the phantom, we can look at how the melanin volume fraction, simulated melanin volume fraction impacts the measured value. And you can see this negative bias as a function of increasing melanin content. Similarly, in photoacoustic oximetry, Bill Vogt has done some excellent work combining an epidermis and dermis layer on top of a breast phantom, and shown that for very low melanin volume fraction, you get a pretty good ability to estimate saturation, but at a very high melanin volume fraction, the signal is very difficult to convert into a saturation value. Okay, so now I'm going to talk very briefly about our most recent pulse oximetry phantom work with Florida International University. We looked at 2 different approaches, one being a very basic molded silicone approach with a 3D printed mold that with an embedded channel, and this could fit into a commercial pulse oximeter. And we had a turbidity in the form of titanium dioxide, and the tube was PVC. And then we also 3D printed some phantoms with channels. We have a couple different designs, one with these U-channel geometries. This is in order to identify what's the minimum channel diameter that the printer could achieve, and it was about 1.2 millimeters. And then we have a more biomimetic geometry with a more compliant photopolymer material, and we were able to combine that with some scatter in order to make a turbid phantom. So we then tested these 2 turbid channel phantoms. And this is using a flow system developed by our collaborator Chris Scully's group. This system shows the oxygen sensor and oxygen tank, so these were not used in this study. We just looked at the PPG signal, and with the 3D printed phantom, we were able to see a fair bit of the PPG signal reproduced using the IR LED. With the molded PDMS phantom, it was harder to discern a pulsating signal, and so we're just kind of going to incorporate a little higher blood content, blood volume within this, and make some other adjustments to improve these phantoms. But this is really just kind of a start, I think, for our work in this area. In conclusion, we believe these pulse oximetry phantoms show a lot of promise as regulatory science tools that can help mitigate racial disparities in performance. We believe that epidermal melanin is a key factor in performance of optical diagnostic devices. This is really shown extensively in the literature, and its effects should be rigorously evaluated in pulse oximetry. But of course, other variables need to be explored as well. Many of the phantom design issues remain outstanding, and so there's a lot of work that needs to be done. Our pigmented epidermis layer phantom from the cerebral oximetry phantom may be effective as a test component for pulse oximetry. And we also think that 3D printing has a lot of promise to advance phantom standardization and improve the quality and ease of dissemination. And finally, I'll just mention that we are hiring postdoctoral research fellows for this project moving forward. Thank you.
All right, Josh, thanks. We have a moment or two only for a few questions. Perfectly clarifying points. We can hold it. We're going to have a wrap up after this too. So there's plenty of time for questions later. We're only, a half hour behind schedule at the moment. All right. I don't see any hands raised. Let's now transition over to Jeff and Veronica, Mike alluded to the fact that his phantom can't detect what wavelength is coming in. It's agnostic to the wavelength. And I think we're going to hear a little bit more about that now. Jeff and Veronica, you are on. There we go. You're sharing. Good.
Can you hear me?
Yes, we can. Can't see you, but we can hear you.
Okay. You can't see the PowerPoint as well at the moment. What's going on with our PowerPoint? You can't see I presume you cannot see my computer screen.
Well, that is correct at the moment. Maybe it's just far away and takes a moment to get here.
Yeah, we're about 8,000 miles away. It's dark and it's raining. And
I also don't see your picture, but you might not have your camera on.
Okay, I'm trying to get the. Can you see any? Can you see the PowerPoint? Can you see my screen now?
Not yet.
No. Because
well, let me ask in the room because it might just be me. Anybody else see?
No.
Okay.
No. Okay, we have two computers going here. One sending it to you and the other receiving it back, so I can see it's not getting through.
Yeah, we can't see it. Fifi, do you have Mike's presentation? Because I think you can display it and Mike can walk through it, or Jeff can walk through it.
We'll see if we can dig it up.
We'll still digging. Let's continue to see if we can get it to work because it did work yesterday, I think. No.
Try pushing the green arrow.
We did.
Oh, are you colorblind? I'm in trouble.
Hello.
Yeah, we're still here. I think Mike is looking to see in his email if he because you sent us your presentation, so he just has to find it.
Yeah, well, I can see you.
Oh, that's good. Oh, wait, I saw something. Whose screen am I looking at now?
Me, I was just somebody's.
Because we were about to come up then.
Yeah, it says we cannot share while someone else is sharing.
Yeah, David, can you kill your share?
Can David stop sharing a minute?
Let's see, can I?
You cannot start screen share whilst the other participant is sharing.
All right, I am God, apparently, according to Zoom. There we go.
Yeah, you can see it now.
Yes, we do.
We're on. We're on.
Please continue.
Jeff, I'm sharing your slides. I will advance for you. Just tell me when you want to advance. Please proceed.
Yeah, we're all right. Go for it. Thank you.
Okay. I don't know if I'm looking at Mike's own. Anyway, so first slide, lab studies do not account. And pulse oximetry should be as accurate as possible for all populations. Are we on that slide? Yeah. Okay, so Jeff and Veronica here. We've been in pulse oximetry for 35 years. The last 30 years have been focused on using spectrometers that check the accuracy and calibration of sensors in particular. Our products are calibrators and they're not simulators. That's background to what we've got to say tonight. Okay, next slide. Our vision of the future of pulse oximetry must not be compromised. We do not accept pulse oximetry is of little use because of accuracy issues. We do not accept limited statistical analysis, which conceals accuracy issues. We do not accept the problem of melanin is not worth fixing. We do not accept manufacturers denying sensors are not as accurate as they used to be. And we do not accept education of end users and the use of pulse oximetry exempts pulse oximetry from being accurate. Next slide. We believe that manufacturers should be compelled to manufacture sensors at least as accurate as they used to be and as accurate as they claim. Statistical analysis should better represent accuracy. Melting bias can be fixed and must be fixed. Pulse oximetry should be as accurate as possible and therefore of use for all populations. Are we missing one off the bottom? No, that's it. Next slide. So there's an issue with sensor accuracy. The number of inaccurate sensors in use from both OEMs and non-OEMs has skyrocketed in recent years. Next slide. This deterioration in sensor accuracy started with the appearance on the market of non-OEM cheaper copies that seem to work and function as well as the OEM product, but many were not as accurate as the manufacturers claimed. Next slide. Many customers moved to these products unaware of accuracy issues. In order to compete with the ease and price, OEMs had to do some cost cutting, and the cheapest supplier got the business. Next slide. The main target was LEDs, as wavelength measurement in mass protection is costly. The result has been a widespread drop in sensor accuracy. Okay. There are reports of high bias in pulse oximetry readings when used in darker skin, dating back 35 years to 1988. Next slide. The emergence of melanin is linked to bipedalism and the loss of body hair in an evolutionary process started 5 to 7 million years ago. The loss of body hair increased the risk of exposure to UV. Melanin decreases that risk. In about the last 50,000 years, humans migrating further north where the UV is weaker, evolved to have less melanin, leading to a range of skin tones. Yeah, and we got to cope with all of those. Next. For a sensor to be accurate, the LED wavelengths must match the calibration data that's being used. That's very clear in ISO. Next. Statistically, if the probe is not on frequency, that might introduce a bias and measurement imprecision in the pulse oximeter. Dr. Pinello said that back in November. The sensor must be on frequency. Emphasize that. If I don't say anything else tonight, that's my message. The sensor must be on frequency. If the sensor is not on frequency, it doesn't matter how much signal processing electronics we've got, because if the sensor's churning out garbage, it's not going to be any good. Melanin can change the centroid wavelength of the red in the sensor as it passes from emitter to receiver. When this happens, the sensor is no longer on frequency. And the other point is the relationship between any sensor accuracy, and any frequency errors is a mathematical inevitability. Okay. Just off the bottom slide, just read it. What's that say? Yeah, erroneous calculation measurements due to inaccurate sensors and high bias caused by melanin. That's the same root cause. Yeah, what we're saying here is a high bias sensor and melanin bias are very similar phenomena. Now here we got a breathe down on 2 OEM Nelcor finger clips, DS100As. One was spot on, and the other had a low bias. So we did a breathe down and compared the two. And the data there with triangles crisscrossing at the bottom of the two is the prediction of the performance of the sensor based on knowledge of the frequency areas. So the prediction made mathematically from wavelength errors and what we saw in the breathe down match very closely. But a comment here, and it's very important. Nelco claimed that every Oximax sensor is uniquely calibrated. This is false. Every reusable Nelco Oximax sensor that we have studied has the same calibration data, regardless of the LED frequencies, often resulting in inaccurate sensors. If this sort of thing is not fixed, anything we do to try and resolve the Mellin bias is going to be swamped by this sort of practice. Okay, next slide. So the magnification of errors, we've seen evidence of this in a few slides recently. The point is, for any fixed spectral error in the sensor, as the SATs fall, the impact on the SATs gets greater. So, for example, here, if you look over the left-hand side of the graph, where the reds are, the lowest graph represents about 97% SATs, then up to 90, 80 and 70, but the point is that gradient is increasing. So any error on the x-axis there, the effect on the y-axis is multiplied. So yeah, that's why we get larger errors at lower SATs. Next slide. So this is what we generate. I suppose this is what clinicians generally think about in terms of sensor accuracy, plus or minus 2 all the way down. Next one. Can you see that one? Can you see that one, Sandy? No.
Yes, we can see it.
Okay, it's coming up very dull here. Okay, there's
Click again.
Where?
Can you click the slide on a bit more? Thank you. And one more. Thank you.
Okay. It's not clear what's going on my computer, but there's a high bias sensor illustrated at the top of the graph, showing the magnification of errors as the SATs fall. So the point is, when the think parallels we want to draw here, we saw data from high bias melanin sensors yesterday. It's very reminiscent of this sort of thing. Next one. So here we got a sensor with a low bias. This is coming out on my screen. You can see what's there.
Click.
So can we click again? So with low reading sensor, the pulse oximeter can read 90 when the patient is at 95.
The other thing to note on those graphs is that with a high reading sensor, the trend is minimized, and with a low reading sensor who's got a low bias, then the trend is exaggerated as people's oxygen is going up or down.
Okay, in the real world, sensor accuracy bias, the number of inaccurate sensors not in frequency and use from both arms has skyrocketed in recent years. Surveys of sensor accuracy show a trend of decreasing accuracy. So 2007, we got 17% failed with a sample size of 1,008 sensors. In 2013, sample size of 758 sensors, 22.3% sensors failed. And more recently, in 2020, a survey of non-OEM sensors had a failure rate of 50%. This is the data from one OEM. In 2018, we saw getting on 25% on the fail in accuracy. So the number of inaccurate sensors not on frequency in use, it's in line with what Bob Kapotti was mentioning back in December. They were saying when they pulled a load of data together, and it looked so grim. You wonder how you could ever use pulse oximetry. Next. So racial bias and inaccurate sensors have the same root cause. The sensor is not in frequency for the R curve in use for which it's to be used. None of these problems will be fixed until there's better control of sensor calibration during manufacture. The Quality replacement sensors, quality of calibration replacement sensors is not always the same as the sensors used to gain regulatory approval, i.e. the offer frequency not accurate. Next. Auditors, either external or internal to the manufacturer, are not currently checking that the sensor is accurate as required, due to the widespread belief that sensor accuracy can only be checked by a desaturation study. Auditors could request that a sensor is calibrated properly on frequency by use of a spectrometer. Prior to 2013, there was no regulatory requirement for pulse oximeters to take dark skin into account. Then in 2013, the FDA required 15% of the population used to generate the calibration data to be darkly pigmented. Next. So that might work for patients who could be thought of as like 15% dark skin and 85% dark skin. But otherwise, you know, this really doesn't address the issue. Next. Oh, it's there. Okay. I can't see it on that slide, but I'll read it out from another slide. So even the calibration data, then we got that one, haven't we? Perhaps even worse is that the 2013 requirements are not retrospective, and pulse oximeter technology already on the market has not had to comply with updated standards and continues to be sold. Next. Melanin. We're of the opinion that whilst melanin is the cause, it's the effect we should be looking at. We're of the opinion that we should be looking at the impact of melanin on transmitted red wavelengths. Next. For example, you know, if the melanin shifts the central wavelength by plus 6 nanometers, that can be measured without having to know how much melanin is present. You just think of maybe shine 663 nanometers into a finger at the point where the pulse oximeter is going to be used and see what comes out the other side. We've done a bit of work on this, and we've seen evidence that the wavelength is shifted upwards. So again, our point here is that I think we should be looking at the effect of melanin rather than trying to quantify dark skin and tannin and lighter skins and things like that. Okay.
So what we're thinking is that manufacturers have already got detailed specifications and accuracy information in their quality system. And perhaps more detailed information should be part of the confidential information provided for regulatory purposes and for compliance checks. Sensor calibration accuracy can be confirmed by use of a spectrometer to check the wavelengths or what they should be in the design to enable the system to function as it's intended to do. What we're saying is we're checking for introduction of an additional bias into the system. We're not so that the system works as it's meant to. And in order for that to happen, auditors need to know what sensor wavelengths should be, and that information needs to be better conveyed than you'll find in current instructions for use and 510K summaries. And auditors could check compliance if they have the manufacturer use a spectrometer to prove it. So if pulse oximetry was more accurate and was recognized as being accurate, then it would be better implemented. And we see that in both low and higher income countries, and there'd be less need for some gold standard invasive expensive testing all the time. I think it's really important that knowledge of accuracy over more discrete SpO2 ranges would actually help provide greater confidence in the use of pulse oximetry. Because if I looked at an example data for some ARMS plots, which gave an ARMS value of 3.38, and the same data, when it was broken down into smaller ranges, gave a really good accuracy of only 1.38 ARMS at the top end of the range from 90 to 100. And I think clinicians, generally, most people using pulse oximetry are interested from about 85 to 95. And if you're in that range, above it or below it. But I do think it's important that we maintain the whole range, but break it up into smaller sections. The amount that we break it up, I think, is something that would need to be decided. But I do think that's really important to help people understand pulse oximetry better. So if you're doing investigations looking at pulse oximeter accuracy, then the design should include assessment of the wavelengths coming from the LEDs. And I think that's really important if you're looking at real world studies to prevent confounding results. Because when you do clinical trials, you're using, as we've already discussed, small populations, restricted testing. And I think the Equiox study needs to be extended, not just looking at the pulse oximeter model and the sensor type and manufacturer, which are already being recorded, but maybe also assess the sensor in some more detail. Just go to the next, you can look at that if you're interested again. I think we've got to understand and remove the variables causing inaccuracy, and that would enable pulse oximetry to be used and trusted as a full diagnostic tool. And I think accuracy information needs to be clearer for the user, and that would help understanding of how to use it. And we've got to watch out that technical innovations that are brought in to address racial disparity don't get swamped by inaccurate sensors, particularly if the current widespread LED performance is still in place. I think we should extend lab studies beyond the small number of sensors and subjects at the moment, because that, as we've discussed over the last 1.5 days, doesn't really represent the real world. And I think if we're looking at testing systems in the lab, then the sensors should be tested at the extremes of the tolerances that the manufacturer is actually going to use in practice, and that then the specifications must be controlled to ensure that accuracy is maintained. And new standards should be applied retrospectively. Can you click on a couple, please? So new standards will need to be applied retrospectively, because we wouldn't want to leave any poor practice still in place. Click.
Okay, to wrap up then, manufacturers must be compelled to manufacture sensors that are as accurate as they claim. All sensors must be on frequency. Auditors need to know what wavelengths a sensor should emit. And that information needs to be better conveyed than by current packaging of 510K summaries, which is at best vague and at times inaccurate. Auditors could request a check of sensor accuracy calibration using a spectrometer. Where calibration information is coded, then the manufacturer shall be able to read the code to enable identification of the target waves and decrypt the wavelengths. So the point on this one is a lot of third-party sensors that are copying Oximax sensors, third-party manufacturers, mainly from the Far East, will get an Oximax sensor from an OEM, and they'll copy it, but they don't know what the calibration data is on that chip. And then the calibration data is lost. And quite often then, the calibration data that they use is not appropriate for the wavelengths that they use, and the sensors have bias. So maybe this is a way around that from a regulatory point of view. Okay, so this must be retrospective, and manufacturers must not be allowed to continue to sell sensors that are not as accurate as they claim by virtue of their historical clearance. Okay, thanks. We do now.
All right, thank you very much. There were a few quick questions and responses. Bob, you have a response very quickly, please?
Let me make sure that I get this on my screen. And that is, Jeff, I really appreciate what you're trying to say, but I wanted to make sure that I could cover what you thought you heard, and that was from yesterday's presentation. Is this being seen?
No.
Yeah, I did push the share. It's over here.
The green button, Bob.
Yeah. Okay. All right, co-colleagues. We will try. Should be there.
Yep.
Okay. So this was a study that was actually closed in 2002. So it's now essentially 21 years old. But it was a study of 32,000 paired and time paired data points of reference co-oximeter to baby blood gas. And so these are all co-oximetered measurements. And I wanted to be certain between these multiple sites that there was only a difference in co-oximetry performance of less than 0.1 for that. So it's really exceedingly tight in its co-oximetry performance. The challenge with these four pulse oximeters was a degradation in performance once you got outside of this window. And this window is approximately 97% to about 85%. So once you got into high saturations, which are definitely problematic for newborns, or below values of 85, which is essential for some of the cyanotic heart defect babies, it was problematic. And that's where this ARMS exceeded 10. So exceeded the standard. So it isn't reuse of sensors. This is real world studies in a very broad, large sample of patients, but they were all babies, and babies inherently have other issues. But I just wanted to make sure that you understood that that had nothing to do with reuse of sensors. However, Sandy, as you can point out, and if you wish to share the table that I had showed you the other day, where FDA has now, I think, 11 categories for different product codes. 2 of which are on reprocessed sensors.
We're going to go there in the next session, so let's not get there.
We're not talking about reused sensors. We're talking about were any of those sensors checked for calibration.
Yep. Well,
whether you or not, it doesn't matter.
I mean, the data that we've had has been from new sensors, not reprocessed sensors.
Very good, but they're not OEM sensors.
Yeah, some of them are.
Yes, we've showed you OEM sensors tonight, and the reference we made to the Nilco one in particular was Nilco OEM.
And the other one, the other graph of the pie chart showing the change
from 0 to 25
was another of the major manufacturers.
Yeah, a major OEM.
All right,
Sandy,
Mike.
Hand raised. Yep.
Mike.
I was a little confused about the comment about the light going into the finger and being wavelength shifted by the melanin. I'm not sure how that could happen, but also I'm not sure what difference it makes.
Okay, the reason it happens is because the melanin absorbs different wavelengths of light to different amounts. The LEDs are not single point wavelength, they're actually a band. And it's the centroid wavelength which is of importance in producing the signal at the photodiode. Because the photodiode is color sensitive, it just knows how many photons get there. So if you've got something in a pathway that has a differential absorption of different wavelengths, you're going to change the overall shape of the light that's going through. So the centroid wavelength shifts.
Oh,
yes,
I see.
If the red centroid wavelength shifts because there's the disproportionate filtering effect of melanin across that spectrum, it affects the calibration of the pulse oximeter.
It acts like a low pass filter.
Yeah, I see. So you end up with a light whose peak is at a different place after you've absorbed.
Yes,
side together. Yeah. Okay.
We've done some work on it in the last 10 years and we keep on coming to the same conclusion.
All right. Any other questions before I make a few general comments? So clearly there's 2 aspects of the issues that you've raised. I mean, the aspect is one of, if you will, construction and clearance. I'll use FDA regulatory parlance. But there's also a post-market aspect, and typically our standards don't cover, construction standards typically don't cover post-market. That's the job of quality systems and regulators to assure that manufacturers continue to produce product that is not adulterated, which is what the US term is. And we have struggled with that internally to figure out how to do that. But that's not we can't. I can't remember what happened last time, and I don't know what. I know we've looked at it before, and I apologize for not being able to recall the exact situations. That being said, I want to transition. Are there any other comments to throw in at the moment for discussion? Manufacturers, do you have, maybe you could talk a little bit about the way that you maintain specification. I mean, I know you have quality control on your components. Maybe you could talk a little bit about how they're specified and how you're not going to describe what your real wavelengths are because that's proprietary or things like that.
Yeah, sure. This is Jake with Medtronic, and I know, from our end, wavelength is something that we pay a lot of attention to in quality for manufacturing. We measure it multiple times to ensure that it meets specification.
Do you have 100% wavelength check?
Yeah, we do 100. So every LED is measured twice, 100% of sensors.
I can speak also. We also have quality systems in place, and then it's also.
Can you get close to a microphone, please? It's a little.
You speak up a little bit. When the mic's on the ceiling, speak up and it'll catch it.
Yeah, we have 100% testing. We pay a lot of attention to testing of components, emitters and photodiodes. So everything is 100% tested.
Yeah, so this may be a problem in smaller manufacturers or
the major manufacturers. We've been getting this denial for 15 years. It's not good enough. The Alco claim all the reusables are uniquely calibrated. If you look at the calibration data in them, it's the same in every one, regardless of the LEDs.
And if manufacturers introduce new sensors and they change the wavelengths, then unless you've actually changed all the monitors that they're going to be used with, you're not going to be able to have accuracy with them anymore.
Yeah, we've had this denial for a long time. And this denial goes on, there's not some honesty here. We can't fix this Melanie problem.
That's why the regulators and the auditors do need to know what the specifications are when the accuracy of the ARMS and the clinical trials are done. And I think it's important that the wavelengths that are used
and that can be confidential.
That can be kept confidential, but that the tolerances for those wavelengths are tested.
Sandy, you got to have
testing the LEDs at the extremes of the tolerance, so that you know that if you have a sensor which has a wavelength on the edge of the tolerance, the manufacturer states it's going to use, that it's still an acceptable accuracy level.
Jake, did you want to say a comment?
Yeah, I was just going to say, the calibrations can be tuned to wavelengths for the sensors. And so if you're picking up a sensor and they all have the same wavelength, you would expect similar calibrations. And so all of that has to be taken into account when you develop a sensor. And especially when we make sensors, if we change wavelength, that's going to be something that we'll recalibrate. We run studies. We take a lot of care for it.
Surely that would need a new 510K, because if you're introducing
correct. Yeah, that's correct.
Curves, and so that should then go through the 510K process in clinical trials.
Correct.
In the US, yes.
So the problem, so many OEMs that churn out sensors, but the wavelengths do not match the calibration data, whereas they used to. So it's possible.
You know what those wavelengths are then?
Yeah. We know.
There's two hands up, Phil.
I apologize. I probably missed this, but have there been real world studies that actually show that sensor degradation increases errors? If so, I apologize for missing that.
The sensors we're talking about are mainly new out the packet. manufacturing problems.
Okay, I get that. Then I would add that in the hypoxia lab, we test a lot of reprocessed sensors. I mean, we've done it for years and years.
These are new sensors, they're not reprocessed.
Well, ones that are repackaged, they're collected after clinical use. They're sanitized. They're repackaged. The adhesive components are replaced. And I can't say that we've noticed any degradation in performance in those items. So at least in that narrow context, we haven't noticed a big problem. I'm not saying that it can't occur in others, but that's just what we've seen.
We've noticed enormous problems with sensors from the Far East, particularly when they copy the NILCO calibration data without knowing what's on the chip, and you put any LEDs in. But on the other hand, we've seen a lot of big OEMs in recent years deteriorate in quality. So they never used to be that 15 or 20 years ago. We've been collecting this sort of data for 30 years. And since about 2005, a lot of them have gone down the pan.
Well, I think that this can be, we should keep this on the front burner for consideration. Unfortunately, right now we are out of time. We still have another topic to cover. Veronica and Jeff, we will try to keep this in the front. I know when people are, particularly around the world, talking about making large numbers of sensors for different types of environment, this might be an appropriate way to verify that they're making a quality product. So it's certainly an important topic, and we'll keep it at the forefront. I did want Paul.
Quick question, Sandy. Okay.
David, I'm sorry, I'm going to have to not call on you. I apologize, because we are definitely out of time. But I did want to call on Paul Batchelder just one time to show just some of the, not confusing data, but some of the data that seems to show that in well-perfused, idealized studies, we don't see large biases as a result of pigmentation. So I just want to make sure that that's in the record and we can study that, because we're all assuming that pigmentation causes all kinds of problems, and I just wanted to for completeness to show some other data.
Okay. What I'm talking about here is based on a large set of data, 195 subjects that were compared to co-oximetry, 43 deeply pigmented and 152 lightly pigmented. The sensor site may not be the correct site for the pigmentation measurement. We can probably gather a lot of information by measuring pigmentation at the sensor site, but what we have seen, we looked at a study that included 8 different studies with 15 different brand or types of sensors. They were all finger clip, and what we saw was a difference in.
Paul, I'm going to pause 1 sec. How long is your presentation? Because we have presenter 2 minutes.
2 or 3 minutes.
Yeah. Okay.
Yeah. So the forehead sensor was compared to these finger clip sensors, and what we see here is the bias between the lightly pigmented subjects and the deeply pigmented subjects in the forehead with the forehead sensor, and the same thing, lightly pigmented to deeply pigmented in the finger sensors. Now the fingers in all of these subjects were much lighter in pigmentation, while the forehead sensor was applied to an area that's much darker. We would think if melanin were an issue here, we would see a significant difference in the bias, and the forehead sensor should, based on theory that melanin is the problem, see the poorer performance with the forehead sensor as compared to the finger sensor. Now, these are all in the same subjects. So forehead and finger in the same subjects. The ARMS for forehead was, the difference was 0.5 between the light and the dark. And the difference in the finger sensor
was 0.8 difference between the forehead and the finger, while the bias with the forehead was much lower than the finger as well. That's really all I wanted to put into the information here. We could talk about it at length, but the bottom line is with empirical data in a large data set, 195, 43 of those being deeply pigmented, the forehead sensor performed a little bit better than the finger sensor, which was much lighter in pigmentation on the same individuals.
All right, Paul, thank you very much. I apologize for hijacking and sticking you in there. Mike, you want to take it away for the next session?